Good morning, students. Today we will discuss another activity for the separation of different constituents of a mixture. This technique is named as chromatography. So first question is, what is the meaning of chromatography? Chromatography is a combination of two words, chroma plus graphy. The meaning of chroma is color and the meaning of graphy is writing. It means to write color. But as we are doing activity, so here the meaning of chromatography is the separation of different colors. Firstly, this technique was used for the separation of different components of a dye, because a dye is a mixture of different colors. So here you can see that the circular spot, this is the spot of a dye, and after this performing this activity, the different colors of dye appears or get separated. That is why this technique is named as chromatography. What is the need of chromatography if we have already different methods of separation of different components of mixture? Actually, this technique is useful or important as compared to the other techniques when the given sample is present in a small amount. That is, this technique is used for the purification of impure sample if it is present in a small amount. Now next is principle, सिद्धांत. कौन से principle पर this technique works? This technique works on the principle that different components of a mixture are adsorbed at different rates on a suitable adsorbent. Now, what is adsorbent? What is adsorption? You know about absorption. In absorption, molecules go inside a compound or the material. For example, sponge. Sponge soaks water, it goes inside. It means the water molecules going inside. If I am taking the example of water, it means water is going inside the sponge. This is absorption. Molecules, ions or atoms, whatever we have the case, they are going inside, then it is absorption. But when molecules, atoms or ions are accumulating, or you can say that binding themselves on the surface of a compound, then it is known as adsorption. In this case, this compound on the surface of which molecules are adsorbing, this is known as adsorbent. This is known as adsorbent, and molecules are known as adsorbate. I hope so. Now you are able to distinguish between adsorption and absorption. Now there will be two phases in this activity. One is stationary phase, which will not move. In this case, our paper, whether we are using filter paper or chromatography paper, is different paper which is used for this activity. But you can also perform this activity with the help of filter paper. Then second is moving phase. Moving phase will be that phase which will move on stationary phase. It means stationary phase is just like path on which our mobile or the moving phase will move. The moving phase will be that solvent we will take to perform this activity and the components of the mixture which will move on our filter paper. So I have already told you that you can even perform this activity with filter paper at home. So, to perform this activity, you will take a filter paper, you will mark 2 centimeter from the bottom, and there you will drop spots of, either you can use your sketch ink or the fountain ink. Okay, then you will allow that filter paper to dry. Then you will take in a glass, it will be a small quantity of water. Uh, why? Because we will not drop the whole filter paper into water. What we will do, we will take only, for example, we will take water up to this level. Okay, then you will insert vertically this filter paper inside the jar or the glass that you have taken, but keep in mind that mark should be above the surface of the water. So what will happen? After some time you will observe that there will be separation of different colors. For example, in this case, on the top it is sky blue color, then it is light purple color, and below it is dark purple color. So different colors of sketch ink get separated. So you can see in procedure also the same procedure will be followed, that you will take a strip of filter paper, then you will mark 2 centimeter from the bottom, then you will drop marks of ink. After that you will allow that strip to dry, and then you will keep that strip in a jar containing solvent. You can take water, or if you can also take alcohol, acetone. Acetone is the nail paint remover. After that you will take out that strip, you will allow that strip to dry, and you will be able to see the separation of different colors. So the last is given in your chapter, uses. What are the uses of chromatography? This technique is used in the pharmaceutical industry, that is to separate different components of the drug. Then it is also used in the cosmetic industry, because chemicals are also used in the cosmetics. Then it is used, this technique, chromatography, is used in the dope test. Dope test means to separate drugs from the blood. Doping test of athletes. This technique is used for the separation of drug from the blood. Then to check contaminants in food and water, chromatography is used. So it was all about chromatography. Thank you and have a nice day.